everyone, this is Mrs. Mallard here, and this is Unit 6, Lesson 5 on meiosis. So the goals for this unit is to understand what cells in our body undergo meiosis, and also to understand the outcomes of meiosis, to understand that we get 4 cells at the end of meiosis, and also understand how genetic variation occurs in the cells that we get at the end of meiosis. So just a quick review, we've already learned about the cell division process called mitosis. And this is the cell division process that makes what are called somatic cells or body cells. So cells in your skin, lining your stomach, etc. These body cells are going to divide through the process called mitosis. And we've already learned about the different phases of the mitosis: prophase, metaphase, anaphase, and telophase. So today we're going to be learning about a different cell division process, but before we do that, we need to kind of front load some vocabulary terms. So the first vocabulary term that we need to talk about is the term homologous chromosomes. So that root term homeo means the same. So if you see like this kind of root in a word, it's referring to the same. So homologous chromosomes are chromosomes that have the same genes. They are the same size, the same shape, all right? And they carry the same genes, but the version of the gene could be different. So here's an example. I have 2 chromosomes. These are homologous because they are the same size, they're the same shape, and they both have a gene for flower color. All right, so both of them have a gene for flower color. However, if you notice, one chromosome has the version for a pink flower and the other chromosome has a version for a white flower. So even though they both have a gene for flower color, it's slightly different between our chromosomes here. So these are called homologous chromosomes. Also wanted to point out before we get into meiosis that humans, we have a total of 46 chromosomes in our cells. And 23 of those chromosomes comes from the father's sperm and 23 comes from the mother's egg. So you can see 23 plus 23 gives us a total of 46. So if you notice, we have a chart over here that kind of has organized and laid out the chromosomes in a nice organized fashion here. And what you'll notice, for example, we have 2 number 1 chromosomes. One of these is going to be from the mom. One of these is going to be from the dad. If we looked at the number 9, the chromosomes at number 9, once again, we get 1 from the mom and we get 1 from the dad. This is a homologous pair of chromosomes that we're looking at on this chart. So now that we've talked about what homologous chromosomes are, we're now going to be able to get into the process of meiosis. So just like mitosis, meiosis is also a cell division process. The main difference, though, is that meiosis is a process that makes gametes, also known as sex cells. So sperm and egg are the 2 cells that are going to form through this process. Remember, all of the other cells, all of the other body cells, cells that make up your skin, your blood cells, cells that line your stomach, those are the other process called mitosis. So meiosis is only used for making sperm and egg cells. Now, one little trick that I use to remember the difference between the 2 is I remember that sperm and egg made me. All right, so that's maybe a way that you can help distinguish meiosis from the other process, mitosis. So for females, the process of meiosis is going to be happening in the ovaries. Ladies, you release 1 egg per month. And so that process of releasing that egg is going to happen once a month in your ovaries. For our gentlemen, meiosis is happening in the testicles to produce sperm. And guys, you're producing 1 million sperm cells per day. Here are the different stages of meiosis. We're not really going to go into all of the phases like we did with mitosis. I put this up here just to show you that we have some things that look familiar: prophase, metaphase, anaphase, telophase. But if you notice, we go through pro, meta, ana, telo 1, and then we go through a second series. So meiosis goes through more stages than mitosis. What we're going to do is just kind of focus on the highlights and not go through each individual stage. So one of the highlights we need to talk about is what happens in prophase 1. So we already talked about homologous chromosomes. All right, remember homologous chromosomes, these are chromosomes that are, they have the same genes, they're the same size. Remember, we get one from your mom and you get one from your dad. What happens in prophase 1 is that these homologous chromosomes come together to pair up, but because they are so closely intertwined and together, what sometimes happens is sections of the DNA on the chromosome get switched between the other chromosome. So we get sections of DNA that get switched. So here's a nice illustration. We have our orange and our maroon chromosome. They come together. They're quite tightly connected and intertwined, to the point that if you notice here, we have a section, the section of our maroon chromosome that got switched to our orange one, and a section of the orange that got switched to the maroon. So visually, you can see that DNA got switched between these two. And this process of crossing over is one of the reasons, or one of the contributing, or one of the main factors why every single sperm cell and every egg cell that is made is genetically different, because at this point, we've switched up the DNA between chromosomes. So this process is called crossing over. The other process that contributes to genetic variation is called independent assortment. All right, so let's take a look at this word, independent assortment. So if you are sorting things, what that means is that you are separating. All right, so if you are sorting things, you are separating items. So in this case, the item that we are sorting or that we're separating out are chromosomes. All right, so that's what we're sorting, is we're separating out chromosomes. And you can see, I've got a picture here, the X's represent chromosomes, and you can see how we're sorting them into two different cells here. And then at the end, they continue to get sorted into these 4 cells. All right, the independent part comes from the fact that one set of chromosomes is going to sort and line up in the middle of the cell randomly or differently than the other set. So what I mean by that is that if you notice, we have 2 sets of chromosomes. Notice in this first one, I've got the red on this side and the blue on this side, okay? Notice the cell kind of towards the end here, I have it flip-flopped. All right. So there's basically 2 different arrangements of how that first set of chromosomes can line up. And so notice this is different than here. Same thing with our second set. I've got a blue one here and a red one here. But if you notice over here, those chromosomes have lined up differently. So this set, that first set, is going to line up independently from the second set, okay? All right, another way of saying that is that when the chromosomes line up in the middle of the cell, they line up randomly, all right? The reason why this is important is because it allows us to get lots of different possible combinations, all right? So we get lots of different possible combinations because of this. So every egg that a woman produces is 1 out of 8 million possible combinations. And every sperm a man produces is 1 out of 8 million possible combinations. So if you think about it, you have to have 1 egg and 1 sperm to create a fertilized egg, which eventually would develop into a fetus. If a sperm is 1 out of 8 million and an egg is 1 out of 8 million, then a baby that is produced is 1 out of 64 trillion possible combinations. This is just another example of how those chromosomes can arrange differently. So this is showing like the two different possibilities of how those chromosomes arrange themselves. All right, so kind of two things I want to emphasize again is that process called crossing over, where the chromosomes switched pieces of sections of DNA, and independent assortment. Those two processes are super, super important because these processes only happen in meiosis. And these processes are the reason why every single sperm and every single egg that is made is genetically different. And another way of saying genetically different is we use the term genetic variation. So sperm cells and egg cells that are produced through the process of meiosis, they all have genetic variation, which just means that they're all genetically different. The DNA in each one of those is just going to be different from each other. So let's go ahead and take a look at what happens at the end of meiosis. At the end of meiosis, we end up with 4 cells. All right, we end up with 4 cells and not 2 like we did in mitosis. The 4 cells are going to be gametes or sex cells, so either 4 eggs or 4 sperm. Each one of these cells is going to be genetically different. So this picture does a nice job of kind of color coding the chromosomes, and you can actually physically see based on color that each one of these cells is different genetically from each other. The other thing I wanted to point out is that the cells that we get at the end of meiosis are going to have they're going to have half the number of chromosomes as the original cell. So most of the cells that we find in humans, okay, majority of our cells have 46 chromosomes. However, sperm cells and egg cells are going to have half the number, which means sperm cells will have 23 chromosomes and egg cells will have 23 as well. So another way of saying half is we would say that the cells are haploid. Haploid just means that the cells have half the number of chromosomes as the original cell. So let's just kind of recap. We learned about mitosis and we've learned about meiosis. So let's just kind of compare and contrast the two processes. Both of these processes are similar in that they are cell division processes. So both processes create new cells, and it's a cell division process. Mitosis, however, is focused on creating new body cells, whereas meiosis is for creating gametes like sperm and egg. Mitosis, we end up with 2 cells at the end, whereas meiosis, we end up with 4. At the end of mitosis, all of my cells are genetically identical. That means the DNA in all of the cells will be the same, whereas at the end of meiosis, the cells are genetically different. Remember, crossing over and independent assortment, those create sperm and egg cells that will have genetic variation. The last thing is that cells that are created in mitosis will have the same number of chromosomes as the original cell. So for example, if we're trying to make skin cells, we're going to have skin cells that divide through mitosis. The chromosomes, the skin cells will have 46 chromosomes in each cell. Whereas with meiosis, we're having the number of chromosomes. So our sperm will have 23 chromosomes, or our egg cells that we make will have 23 as well. So this is a nice comparison and contrasting of the two processes. And this right here really kind of breaks down the essentials that you need for comparing and contrasting these two cell division processes.